Okay, good afternoon, everyone. Let's get started. So in the last class, I think we were discussing about diabetes and different approaches. So in today's class, we will see those. And in yesterday's class, we talked about, I think in the last class, we talked about type 1 and type 2 diabetes. Type 1 diabetes is because of lack of deficiency of insulin, whereas type 2 is resistance to insulin. So basically in type 1, there is some problem with beta cells. And as I was discussing in the last class, there could be multiple deficiencies, multiple problems with that could affect beta cells. So something like viruses or obesity or excess body weight, and so on, many things, even insulin resistance also. I hope you remember when I spoke about type 2 diabetes. It happens in two different phases. One is first one is insulin resistance, and that is followed by insulin the apoptosis of beta cells, because they are overproducing insulin. And so many other things, including pregnancy or puberty, during gestation and so on. Many things can happen. And also, there are many other chemicals that could influence the beta cells, because of which the insulin production could be reduced. So we also discussed about what are the problems with hyperglycemia, that is the high percentage of blood glucose, and it could affect many organs, resulting in diabetic nephropathy, diabetic cardiomyopathy, diabetic retinopathy, periodontal disease, and encephalopathy. So diabetes is like a slow, gradual, multi-organ failure. It is something like that. And one that we know very well is about the diabetic foot. It's about resulting in amputation when there is, because the peripheral blood vessels are they narrow down and their nerves are not functioning well, because of which there is less and less, increasingly less supply of blood to the extremities like toes and so on. So there might be a sudden blockage, because of which the whole tissue might die out. And then there is no option but to amputate that, and then try to see if the patient can survive longer. So there are several symptoms for low blood sugar as well as blood sugar. I think we are very unlikely to have low blood sugar, because we even have our secret stash of chocolates also sometimes, isn't it? Anyway, we need to take care of both. We shouldn't have less or higher, because if you have less, you cannot perform your daily actions properly, and eventually it is going to affect the brain and so on. If it is high blood sugar, there is frequent urination, because the kidney is trying to clean up all the glucose, and along with glucose, you would also lose water. So there should be a lot of, because you are urinating, you have to drink a lot of water, and you feel extremely thirsty because of that. And we are usually drowsy, and we have many other problems associated with that. So in today's class, the focus is on what to do in case of diabetes. There are multiple ways, and one of it, we will see a few of them. First one is how to manage it in the early stages of diabetes, or if we know that our parents and grandparents all have it, then we should have a plan of management. There are medicines available, the modern evidence-based medicines. Then we have recombinant insulin to supplement the body with extra insulin in case of type 1 diabetes. And there are stem cell based approaches, which we will only see very superficially. And then there are cellular and genetic approach. This is what is the actual part of it. And we'll also discuss a little bit about recombinant insulin while we learn this. So the first thing is management. The type of diet we take, they should have low glycemic index. which would mean that as soon as if you take 100 grams of food, and right away it is all converted directly into glucose and increases the blood glucose, that means it has high glycemic index. If you have taken 100 grams of some type of food, but it is not digested immediately, it takes a while to produce the glucose, that is kind of has a low glycemic index. So it's always better to take food that has low glycemic index. It's not that we should completely avoid carbohydrates, but we should not take more than required. If one is taking a typical vegetable, I think vegetable as just cooked only vegetable, I'm not talking about the rice and all, it has proper mixture of all the things, proteins, carbohydrates, and lipids, and so on. Maybe not so much of fats, but still. But if you take rice, it's most of it is only carbohydrate, and we take a lot of carbohydrates, including me. That's the problem, and it has a high glycemic index. Idli also is high glycemic. I mean, carbohydrate-rich foods, especially those are polymers of glucose, usually tend to have high glycemic index. So we should reduce the amount of those and increase more of protein in our diet. And we should eat less and less frequently. And there is also something called as calorie restriction. That means sometimes, occasionally, it's also a good thing to fast, maybe at least one time a day. I mean, one half of the day. It's also occasionally weekly once something. Fasting is also a good thing. But in our daily schedule, say 6 days, if you are working and if you are going to work with while you are fasting, it's actually going to cause a lot of problems with the profession or you as a student too. And Sunday is the only day we get to relax, and probably if we steal that, then it's a lot more troublesome. So we need to find a balance about what we can do or how we can manage. The other thing, as I was mentioning last time, one should have enough exercise, have a very active daily life. Active in the sense, throughout the day, we should have movement during the day, and thrice a week, at least thrice a week, we should have good body workout, jogging, playing games, and so on. If you can do it every day, that's also fantastic. So there should be balance between how much how much are we taking, how much we are working out. And as I'm saying all these things, it also depends a lot on our genetics. If we are already genetically predisposed, or we have other kinds of issues like obesity, or many things can happen because of genes and epigenetics. So, but then you have to probably take professional help in case you really want to, you feel that there is something wrong. As young adults like you, it's not so much of a trouble. But say from 35 onwards, one has to take care of all this stuff, especially if parents and grandparents on either side have these issues. So about insulin, we will discuss in a while. The next one is about to try with medicines. And the last class I did show you these. There are some medicines like TZT, which can reduce the release of free fatty acids and increase the uptake of glucose by muscles and so on. I like this HEI. It's a glucosidase inhibitor, which will reduce the uptake of glucose. I think we cannot stop eating, and if there is a way of reducing the uptake of glucose, I see it as a good thing. But as I said earlier in the last class, any type of medicines, medicines we take that are effective do have side effects. Okay, some have immediate side effects, some have long-term side effects. In case of diabetes, the medication is usually too long. Say it takes several years. I mean, you have to keep continuing it. Actually, it's managing your body that way. So one has to keep track of those things, and professional help is always important. Consult your doctor, and then they will take care of those. So, but that's about medicines. The point is to reduce the blood glucose. So this is blood glucose. There is something is coming into the blood glucose. How to stop glucose into the blood, how to reduce the glucose into the blood, how to remove the how to remove the blood glucose, how to reduce the inflow of glucose into blood. Those are general strategies for these medicines. Again, I think there are a lot of purported medicines by alternate medicines like homeopathy, Ayurveda, and all those things. I think one has to exercise caution, try to know the biology. We as biology students are different from many other people in the sense that we are able to follow the literature. We can find the literature, we can read the literature and come to a judgment about what type of medicine we have to take. I mean, I myself take, say, turmeric or tulsi leaves and some things like that. Okay, my grandmother told, so I do take those stuff. We have bias for our own traditional medicine, right? We want to believe that it is true and right. But while I am also biased, I think it's important to, when there is a serious issue, as long as it's not a serious issue and whatever you're doing may not affect. But if it is a serious issue, and your approach should be very clear. I'm saying this because we are biology students and we can understand. So try to read before you do something. Say, if they say biguanide inhibits the release of glucose from liver, we should have known exactly what this particular compound, to which molecule it is interacting and which cells are responding. We should have known the mechanistic details of the approaches that we actually choose. So be unbiased, okay? Yeah, be objective. That is the point of it. There are thousands of traditional medicines all over the world, and we would only like our traditional medicine because it is ours. We don't go for everybody else's traditional medicine, isn't it? So we should not fall in trap of our own biases. So that is what I'm trying to emphasize. Okay, but yeah, evidence-based medicine or the modern medicine that you say, or allopathy, they are effective and they do have side effects, and that's why professional help is required about the dosage, when to take, when not to take, how long to take. Okay, but that is about evidence-based medicine. Okay, so there are also stem cell-based approaches in the sense, say, if it is type 1 diabetes where the beta cells are deficient or they're dead and so on. In that case, if we can have some cells that can take the function of these beta cells, then that is a fantastic approach to do that, isn't it? Because they would know when to express, how much to express, and how to shut it out. Okay, and we don't have to, and it is unlikely that there will be any side effects. But these are medicines of these are approaches for the future. At this point, there is still in clinical research, maybe it's in the mice, not even in humans. But the general approach is to take, say, there are 3 approaches here. One of it is to take the endoderm and coerce or induce the differentiation using biochemicals factors in the growth condition to produce insulin expressing cells and put it into the patient. That is one of the approach. The other one is using induced pluripotent stem cells. First get induced pluripotent stem cells, then differentiate them along to become insulin expressing cells and put it into the patient. The third one, I mean, still people are working on it, or nothing is confirmed at this stage about stem cell technology. It is a medicine or medical approach of the future. Majority of the regenerative medicine is for the future, because we still have a lot of issues, including ethical issues. We also have issues of technical and the expense as well. So we can also take mesenchymal stem cells that are very easily available, and if we can figure out what factors can differentiate them into insulin expressing cells or cells similar to that of beta cell, then it will be a good success. So it's all about getting the cells and putting it into the patient. While I say it very easily, putting it into the patient, it's not that easy, right? It is produced in the pancreas, islets of Langerhans, and it should be, there are alpha cells, beta cells are what we're trying to put, there are delta cells, and so on. The cells we put in should integrate with the rest of the cells, isn't it? Only then they will be able to survive in the niche and be and function as if they were beta cells. Okay. Yes, those are the approaches that using stem cell technology. But then, as I was telling, if you really differentiated beta cells and put, the first problem is that we already have immune issues, right? First of all, how did we lose the beta cells? How did the patient lose the beta cells? It could be because of the antibodies against beta cells, okay? And even if you differentiate new beta cells and put, they will also be attacked. That is one of the problems. And if you take cells from somebody else and put it into one patient, there is immunorejection. And we don't know if we put some cells like beta cells, how long they would survive. So there are multiple questions. That is why I maintain that the stem cell technology approach is for the future. We have so many things to work on. All these are the problems. There is a risk of infection, ethical issues. We don't know if you are making any changes in the genome that we will discuss in the later class. Then there is also another problem. There are endogenous viruses, many other things. Okay, so people are struggling, each one in one or more problems. So if we even take stem cell approach or any other approach, we have several things. First thing is we should have insulin expression. The cell should produce insulin. Second thing is the insulin produced, insulin expressed should be processed, which we will discuss, you already know, I guess. Then it should be able to time the production of insulin, when it should be produced, when it should be shut down. Okay. Then we want it for a long time. It should be, the cells that we put in should be available for a longer time. They should not be dead in sometimes. That's what these two would mean. And there should be absolute incorporation of the introduced cells with the rest of the body, rest of the immediate tissues and the body in general. Okay. Okay. The insulin is structure of insulin. I guess you know that there are two chains, chain A and chain B. And there are two disulfide bonds between these two chains. So there are the chain A is 30 amino acids and chain B is 31 amino acids, a total of 51 amino acids. It's kind of one of the smallest proteins you can tell of. Smallest proteins with secondary or quaternary structure. While I say that, it's not exactly that's not exactly the way it is produced. It is actually produced as pre-pro-insulin. That is one complete chain like this, one single polypeptide chain. It has a signal sequence that is for protein targeting within the cell during production, during translation. Then we have chain B, and then we have chain C, and then chain A. Eventually, it is the signal sequence is cut off and resulting in pro-insulin. Okay, so there is chain B and chain A and chain C, and there are two disulfide bonds here and one disulfide bond within chain A. And later, the chain C is cut off. Now, until here, it is a single polypeptide chain, but now once it is insulin made, it has become into two different chains, and we are calling it as chain A and chain B. Chain C is deleted out. Okay. The production, if you look at here, I hope you remember the signal sequence is important for targeting. Once if you remember the protein translation in cell biology long time ago, you must have learned. Translation happens on free ribosomes or on rough endoplasmic reticulum. All those that have to be targeted to any of the cell membranes, cell organelles, or should be secreted, are usually translated on top of rough endoplasmic reticulum. So the signal sequence, so ribosome is somewhere here. Signal sequence, when it's produced, it will halt the ribosome translation further, and then the ribosome will sit on the translocon and on the surface of rough endoplasmic reticulum, and it is continuously secreted into the rough endoplasmic reticulum. And then it is folded up, and while it is being moved to these, say there is one cut here, and that is where the signal sequence is lost, and then it moves from rough endoplasmic reticulum to the cis and Golgi complex, and then towards that to trans-cis Golgi complex, and finally will form a vesicle. So in each of the cases, you have continuous degradation, that is, processing that is happening. So carboxypeptidase E is what will result in the removal of the chain C, and now we have completely or mature insulin ready for secretion. So why I discuss this is, if there is a patient and that person is supposed to be given insulin, then we should have a source for it. In the earliest of the days, if we remember, in the earliest days, what they do, the scientists went to the slaughterhouses and they collected pancreas from lots of thousands and thousands of pigs. And from so many slaughtered pigs, of course, the pigs were sold for the pork. And from there, they collected from the pancreas, they tried to collect insulin. I think they must have gotten some few grams of insulin in purest of the stage. They must have tried, if I remember. I don't remember the exact details, because, why I'm doubtful is if we inject into the blood, it might cause issues with immunogenic reactions and so on. So that was first experiments were done trying to isolate insulin from the pancreas. So now, as the recombinant technology has become much more obvious that it is it's a technology of the with high potential, or everything is going to depend on anything we want to treat any animal or any organism, it has to do with manipulation of the genome or genes. That is going to be there as long as we are alive. So that will become an essential part. And so there were several ideas. Now we just knew that chain A and chain B, and there are two disulfide bonds, right? If we want to make insulin, and that too, I think it is called as humulin, to induce human insulin. So we need to have two chains bound by disulfide bonds. And for that, how to make use of it? People have built vectors, and into the vectors, they have put the insulin chain A or chain B, and they produced the chains, and then they try to allow the formation of disulfide bonds. That did work for a long while. I think even now, this is kind of what they do. Chain A and chain B is here. They are in two different cells in vectors, and in one cell, using one cell, they will produce a large quantity of chain A, and then they will make chain B on this side, another set of cells. They will take equal proportions, chain B, equal proportions, and try to make the disulfide bonds between them, and then you have a final insulin. That is what is used. There are possibilities of other things. As disulfide bonds between chain B and chain B may also happen. Chain A and chain A may also happen. So I think people use different strategies, which is mostly dependent on the chemistry, and also about the kinetics of how disulfide bond forms to minimize unnecessary disulfide bond formation. Yeah, after allowing the disulfide bond formation, they should have purified it, and those that have 51 amino acids will be a combination of chain A and chain B. So they would have purified this and then sold it as insulin. The question here is, why did we have this part of beta-galactosidase, a G enzyme? It is a translational fusion in this case, also in this case, if you notice that. If you remember, I think I was supposed to take expression of lethal genes. Please remind me if I forget to do that. I just looked at it and then I recollected that I was supposed to take that as well. So I will do that. Remind me if I miss it some other time. Yeah. So anytime if you have E. coli and you're producing small peptides, okay, smaller ones, they don't fold properly and they are readily degraded by the proteases. Okay. So although you have the vector, everything, the small open reading frame, and you were expressing it, the produced peptide is readily degraded. So translational fusion with something like beta-galactosidase, if you are going to put, this is the chain A, for example, and this is the part of beta-galactosidase, then we are made a translational fusion. It is produced as a molecule, something like this. Then it is stable. Okay, it is not attacked by the proteases. This is, I'm talking about E. coli. So that is the reason why they made it into translational fusions. Then when you say, I want to, the insulin has only chain A and chain B. It doesn't have these parts. So we need to get rid of this part. So for that, you can use many things. Say, for example, we discussed in the last class, you can use enterokinase peptide, in the sense that peptide that can be recognized by enterokinase, or trypsin. No, trypsin is not useful, but you can use self-cleaving peptides, like we have discussed in the previous lecture on piggyback transposons. Piggyback transposon based induction of pluripotency. We can also try out something called as inteins. Have you heard about inteins before? I hope you know introns, right? What are introns? Intervening sequences, right? They're intervening in the cistronic region, so they call it introns, right? Tron comes from cistron, intervening, so you get in from there, and intron. Tron comes from half part of cistron. So similarly, there are, this is at the level of DNA or even RNA. And upon what happens to these introns? Splicing happens, splicing of exons. So exon 1 and exon 2 that are here, 1 and 2, they get, they fuse or splice together like this, and the intron is left out. And I hope you remember also that some of the introns are self-cleaving. They are like ribozymes. They can mediate the cleavage by themselves. Similar to that, people have observed that there are some protein sequences, proteins at the level of amino acids. Okay, these are proteins. They have certain sequences, and those sequences can cleave themselves and make it into two different proteins. It is almost similar to that of self-cleaving peptides. So, sorry, when they cleave, when they get out, I think this is the mature protein. Sorry, I will try to erase it. So the mature protein is something like this. It will form, and the intine, now they call it as intine. In for intervening, tene, this tene comes from protein. Inteins can also be used. Okay, but what I'm trying to imply in this particular section is that what you can now include a sequence here, so that beta galactosidase part and the chain A or chain B would separate, and then using any of the molecular techniques, you can separate them using chromatographic techniques, and then use it for binding, making chain A and chain B, so that full insulin is formed. Okay, that is the approach trying to make recombinant insulin. So this is better than the pig ones because it will not arise any immunogenic or other kinds of issues. It's as normal as a human one. The only problem is how much to give, when to give, how long to give, isn't it? So those are, again, some of the issues that are there. People have worked out the dosage and also adjusted the diet requirements, and the dosage is also dependent on many other aspects of the patient as well. So what we want to do is, before I'll talk about this. We want to have insulin, we have produced recombinant insulin, but the problem is how much to give, when to give, right? Timing of insulin release. How does it happen in normal cells? Normal cells, this is the beta cell in the pancreas, and here is the target cell, usually the skeletal muscle cell or the liver. Beta cell has GLUT2 transporters, so the glucose after a meal is mixed in the blood, which is sensed by GLUT2, and GLUT2 increases glycolysis and ATP. ATP by ADP ratio is increased, which will activate ATP sensitive potassium channel, because of which a lot of potassium will move, and because of that there is membrane depolarization, and that stimulates the voltage dependent calcium channel. So there is a lot of secretion of insulin from the extracellular, lot of inflow of calcium ions from the extracellular side into the cytoplasm. And also there is release of calcium from smooth endoplasmic reticulum into the cell. So this increase in cytoplasmic calcium will mediate the insulin vesicles to fuse with the membrane like this. And then insulin is secreted out into the blood. From blood, it is carried to several all over the body, and the ones that some of the target cells include skeletal muscle cells. Insulin binds to the insulin receptor, and that phosphorylates insulin receptor substrate, which IRS, insulin receptor substrate, that activates PI3 kinase, phosphoinositol 3 kinase, which will increase the, which will actually, where is it? Yeah, there are many other steps here. I think I missed the slide, but I'll put it again, okay? 1 second, sorry. I wanted one other slide. I must have discussed that in the last class. Just wait a minute. Okay, yeah. So this is from CARP, cell biology. You must have read it in your third semester or so. Insulin binds here. It activates IRS. IRS activates PI3 kinase, and that activates PKD1, which will activate PKB and will allow the fusion of cytoplasmic vesicles containing GLUT4 transporters onto the cell membrane. So now there are GLUT4 on surface of the target cell, and they will take up the glucose. So I want to ask a question. Did I show you this figure before as a part of this class? Yes, no? Can someone answer, please? You don't remember? Okay, good. So this is facilitated diffusion. I hope you will, and it is passive in the sense there is no input of energy to take up glucose. So in that case, I'm trying to discuss this so that it will be useful for your thinking in general. Say, assume you have 20 glucose molecules on the surface and there are no GLUT4, then they cannot enter in, right? Glucose cannot move through the cell membrane. But upon stimulation by insulin, like in here, and eventually the GLUT4 moving on to the, or fusion of the vesicles, now you have GLUT4 on surface. And as I said, it is passive diffusion and there is a gradient. You have 20 outside and assume that there are 0 inside. So the gradient is high concentration on the outside and low concentration in the cytoplasm. So assume that 10 glucose molecules entered inside, and then there is 10 here. Now what is the gradient? There is no gradient. It's same, right? And it is not active transport, so there will not be any further movement. It's in equilibrium. But these glucose molecules, through group translocation, are made into glucose 6-phosphate by glucose kinase. Now, all these 10 are made into 10 glucose 6-phosphate. So how many glucose molecules are there then? There are 0 in the cytoplasm. Now there is a gradient again. You have 10 glucose molecules outside and 0 here. So they'll become 5 and 5. And all these 5 will again be made into glucose 6-phosphate. Then there you have 2 and 3, something like that. That is how the blood glucose that is present outside is taken in. Okay? Yes. So yeah, this is how beta cells respond. Here they have GLUT2 and they increase the ATP-ADP ratio because now glucose inside, there is a lot of energy, and then potassium, ATP sensitive potassium channel, voltage gated calcium channel, and release of insulin. Yes. So now we are actually going to read this particular lesson as last time I was telling you about. So I have described a lot of background, and that is sufficient for you to actually understand the lesson, the paper too. But let's discuss why did they choose enteroendocrine cells? And second thing is, how did they make them secrete insulin? Enteroendocrine cells are in the lining of the intestine, and they do not produce insulin. Insulin is produced by beta cells. So we need to understand how they made the enteroendocrine cells secrete insulin. And question is, why did they choose enteroendocrine cells? Okay. And of course, this is to reduce the hyperglycemia or address the question of diabetes. So first question is about why choose enteroendocrine cells. Enteroendocrine cells there are a lot of gastric pits and so on. You see that there are a lot of invaginations which will have different variety of cells, including mucus secreting cells, gastric glands, and so on. In them, there are also something called as enteroendocrine cells. Each of these have several functions, and also enteroendocrine cells also have a function. The function is that whenever upon digestion or so, you can say the glucose that is made is taken in by all the cells. In some cases, the glucose moves from cells to the blood. That is, thereby it is increasing the blood glucose, as we discussed before, which might be sensed by the beta cell. This is in the pancreas. Just for figure's sake, they have put it like this, but otherwise, it is sensed by pancreas, increase in calcium ion, cytoplasmic calcium ions, and release in insulin. Right? There are also other ways of how things happen. This is K cell. This is the enteroendocrine cell that we are talking about. One of the types that they have chosen is that it produces GIP, gastric inhibitory peptide, which binds to gastric inhibitory peptide receptor present on the cell surface of beta cells. So in a way, there are two things responsible for the secretion of insulin. Actually, three. You can talk about GLP-1 also. The beta cell is typically responding to glucose that is coming from the intestine, not because it is broken down from liver, stored glycogen in the liver or anywhere else. That would not be, that is not so required. Say, for example, you are saying the target cell of beta cell or insulin is, largely liver cell and skeletal muscle cell. And in presence of glucagon, also glycogenolysis happens and glucose is produced. And blood glucose now increases. If this blood glucose is to stimulate beta cell, it will produce insulin. And again, it has to work on the same target tissues to take up the glucose. That's not an intelligent design, isn't it? So this is therefore, I think the design is somewhere like this. Beta cell has to get multiple signals from the stomach or intestine to sense that there is glucose. And if these are also produced, they are produced because these cells have taken up glucose. Glucose is in the intestine or stomach, and that is what these cells have taken and produce GIP. Same thing is with GLP. And beta cell is receiving responses from three different things. These two are insufficient because glucose is required to increase the ATP-ADP ratio, right? So when you have all the three signals, what would that mean? That means that all this sugar that is in the blood is from the because of digestion of the food, not because it is the glycogen in the body is broken down. Yes. So why these authors chose K cells? Probably there were papers before this where they have tried liver cells and so on as well. Liver and pancreas are very close tissues in space as well as in their differentiation lineage. Okay, so um, the question here is that these K cells, and they produce GIP. When are they producing it? They are producing in response to the glucose that is just digested. You remember we were talking about we want insulin to be produced or secreted only after a meal. And K cells already have that sensing mechanism. They are producing the they are responding to the intestinal glucose, right? So those are a good candidates, so that they can time their secretion based on the intake of or ingested food. There is a problem, however, is they do not produce insulin, right? The cells are good for sensing mechanisms, but these cells do not produce insulin, K cells. So to address this, this is the same thing what I was talking before. So I'll just skip. So to what they have done for this is they were working with mice. So their system is a little bit different from us in terms of insulin. They have insulin 1 and insulin 2, something like that. We humans have only one. So what they have done is they have taken the vector. Okay, I'll just use this. I'll talk about only this one. They have taken the vector. Here is the vector, which is called as GIP. We will discuss rat insulin and neomycin for positive selection, neomycin resistance gene. Okay. This is truncated cytomegalovirus that is for overexpression of the rat insulin. Okay, here they worked on rats. If I said mouse, that's wrong probably. Or they used rat insulin, the cDNA. I would want you to be competent at this stage to be able to read and figure out what they have done, just looking at the diagram. And because all the things that we have discussed here is evident here. They have constructed a vector for secretion of, for expression of insulin. Okay, and they have chosen GIP. Why did they choose GIP, promoter of GIP? Because they have chosen the K cells which produce GIP. That means GIP is produced when they have only in this particular cell, K cell. It is specific to K cell. So they have chosen a promoter for that particular thing, for timed secretion or timed production or quantity of GIP. And there are two things they have tried. One is just having the promoter alone of GIP, and in another case, they have tried GIP along with the first intron. Okay. Somehow in their results, they found that this one, the second case where the intron 1 was included, had better production of the insulin gene. So it's a better expression. So we don't know exactly all the details. That's not the important point at this stage, but you should know that why they have chosen GIP, promoter of GIP. It is because that particular GIP is secreted, or GIP is expressed in K cells. And then this is the intron part. If they were choosing only GIP, then it would be directly with the rat insulin gene. And then they have a signal for polyadenylation. All eukaryotic mRNAs, majority of them, they have for better expression, you should have a GCAP as well as a poly-A tail. So that's why they have used poly-A tail. They have used neomycin resistance gene as a marker for selecting the cells that are transformed, that are that are transfected with this vector. And they have used ampicillin resistance because they have to construct this whole recombinant vector, and that can be done only in E. coli. So as a selectable marker within E. coli, they have used this one. Okay. And then they transfected it into cells and then checked out if insulin is produced or not. Okay. So if there is a question to you, a condition is given, and you are asked to make or design a protocol of how you want to do, or a flowchart of how you want to address this situation. This is the level of thought we should have employed about choice of cells. Why did you choose that? Because that is the only thing available in our lab is not a good answer. We should have reasoned why that is the best source. Okay. In many cases in our labs, we use what we have because we cannot always purchase what is the best. But then having the logic and being able to explain these things in your reviews and project reviews also is a good way of trying to show that you have mastered what you are doing. It's good for research aptitude. So they have done reverse transcriptase PCR. I hope you remember reverse transcriptase. In the last paper also we discussed difference between reverse transcriptase PCR and RT-PCR, right? Here they just have to show if expression happened or not. Okay. So GLUT2 is expressed or not. These are the cells that we are looking at. GI with insulin 7, and that is where those are the transformed cells. And you can see that the other cells do not have, this is what I will try to focus most on. The other cells do not have any expression of insulin, whereas the transformed ones have good amount of insulin expression. Okay. And they have eventually gone to put it into the person, or in just cells. You can also see that they were able to find out that these cells were producing. So they have used glucokinase. You remember that it is important for conversion of glucose to glucose 6-phosphate, and ABC is for against anti-insulin. So this is anti-insulin antibodies. This one is for glucose kinase. This is for GLUT2 expression. And here you are seeing, here you are only showing it is RNA, right? The RNA is expressed. It doesn't mean that the protein has formed. We should be very clear about what we say when you are saying it. We cannot conclude that insulin is produced. We can only conclude that insulin gene is transcribed. Okay. So protein should be there. So they used, this is immunocytochemical analysis. We don't have to bother too much, but as a proof here, they show that, say, these are the A, D, G. They are the cells that have been transfected cells with the vector they have constructed. And you can see that there is expression of insulin, right? You can see that there is insulin expression. You can also see that they are having glucose kinase and also GLUT2. So they're showing that they can take up the glucose, they can time the production of insulin. And eventually they have put it into the, in cells in vitro they have done, and also in vivo they have shown that the rats that were modified or into these cells, in which these cells were introduced, actually reverse the hyperglycemic conditions. Okay. I'll stop here, and I would want you to read the paper, and all you need within that paper is this important point. This will become the central part of it. why did they choose the enteroendocrine K cells, and what did they do with the construction of recombinant DNA? What are those components, and so on. Okay. If you have any questions, please ask, and I'll try to address.